Thank you for joining us today for this CAST webinar. I'm Kent Chesky and I serve as the Executive Vice President of the Council for Agricultural Science and Technology, or CAST. Very pleased to share with you our most recent CAST issue paper on agriculture and the microbiome. Today's webinar is being recorded and will be available for viewing at the CAST website. As noted on the agenda, we'll have a Q&A session with a panel of our authors from this issue paper after our main presentation. We'll open up the chat box for questions related to this paper in just a few minutes. Next slide, please. CAST was formed in 1972 to provide unbiased, credible, science-based information about food and agriculture to policymakers, the media, private sector, and the public. CAST is a membership-based organization comprised of scientific and professional societies, nonprofit organizations, universities, companies, and many individuals. Next slide, please. It is through our network of members and member organizations that we select topics for CAST papers, as well as access a network of scientists, economists, engineers, and other subject matters specialists who help produce these papers. Next slide, please. CAST has a small staff located in Ames, Iowa. Much of the work that CAST does is through our volunteers who serve on our governing boards, task forces that compile and write our papers, and reviewers who make sure our papers are unbiased and based upon sound science. Over the past 13 years, we have involved over 1,000 subject matter specialists who graciously volunteer their time, energy, expertise to help us accomplish this work. Next slide, please. Our publications and information are intended for audiences of policymakers, the media, the private sector, public, and educators. We do this to increase the accessibility, awareness, and understanding of the science, technology, and innovation that is so critical to food and agriculture. Next slide, please. If you visit our website, you will see that we have several different types of CAST publications. Our most popular are our CAST issue papers and commentaries. We also produce Ag Quick Cast for each of our papers that serve as an easy to access executive summary of the key information. We've recently introduced a student study guide, which we hope will be used by teachers and students to learn more about the current food and agricultural science issues. Listed on our website are the other CAST publications we'll be releasing over the next several months and through the rest of 2020. Next slide, please. The use of social media is a very important part of our science communication efforts, and I would invite you to join us on any of these platforms. Again, a reminder, today's webinar is being recorded. Toward the end of the main session, we'll post a message that the chat box is open for you to submit any questions to our panelists. Next slide, please. Like all of our CAST publications, this paper was compiled and written by a task force of subject matter specialists. The task force co-chairs for this paper were Dr. Ignazio Carbone, professor of plant pathology at North Carolina State University, and Dr. Megan Andrews, project manager at North Carolina State University. Dr. Carbone joined the Department of Plant Pathology at North Carolina State University in 2002. He is a population biologist interested in biogeography, molecular systematics, ecology, and evolution. He is also actively developing software tools to facilitate evolutionary and population genetic analysis. Dr. Megan Andrews is a biogeochemist with 15 years experience in studying how different groups of organisms influence biogeochemical processes. At this point, I'd like to call upon Dr. Carbone to lead us in today's presentation. Thank you.
Okay, thank you, Ken. And on behalf of the task force, we want to thank you and CAST affiliates for the opportunity to address the topic of the agriculture and the microbiome, which is an emerging area of interest to the ag biotech sector, farmers, and the American consumer. Now, before I leap into this, I want to introduce the task force members shown here. In addition to myself and Megan Andrews, we have Andrew Binder from North Carolina State, Natalie Brakefield from Unileaf Symbiotics, Owen Duckworth from NC State, Kirk Francis from BASF, Susanna Harris from the University of North Carolina at Chapel Hill, Linda Kinkel from the University of Minnesota, Jan Leach from Colorado State University, Greg Maloney from Novozymes, Kelsey McLeod from SAS, Elizabeth Schenk from the University of Massachusetts Medical School, Elizabeth Stulberg from the Alliance of Crop, Soil, and Environmental Science Societies, Carolyn Young from the Noble Research Institute, and Cheryl Yu Su from Novozymes. So we really like to thank the reviewers. In addition, Kelly Eversole from the International Alliance for Phytobiomes Research, and Elizabeth Pearson from Texas A&M University. We thank the reviewers for their insightful comments on the document and also edits in finalizing this publication. Can I have the next slide, please? Okay, now why did we decide to tackle the agricultural microbiome? I am the director of the Center for Integrated Fungal Research at NC State University. CIFER is an interdisciplinary team of 17 scientists, shown here on the left, spanning 4 colleges, as shown in the pie chart on the right, with expertise in chemical and environmental engineering, mathematical modeling, microbiology, soil science, evolutionary biology, and ecology. As shown in the network at the top right, we are committed to developing an integrated understanding of the biotic and abiotic interactions in ecosystems and at different genetic scales, all the way from the molecular level to individuals and populations and species. So the interdisciplinary interactions in the center emphasize the need for a cross-disciplinary approach to the agricultural microbiome, which led us to the development of this CAST publication. Can I have the next one? Thank you. So since ancient times, modern agriculture has seen unprecedented enhancements in crop yields, thanks to mechanization, plant breeding, fertilization regimes, and disease management. As you can see in the graph on the right, while yields have increased over the past 50 years, shown in the red line, the amount of cropland per capita, the blue line, has declined over the same time period, as available farmland is replaced by urban development, as shown in the picture on the left. Now, continued population growth, the green line, which is projected to hit almost 10 billion by 2050, requires increased yields and better utilization of existing land. Next slide, please. So shown here is a timeline of agricultural revolutions and scientific advances. Starting with the invention of the tractor in the late 1800s and the green revolution in the 1960s, farmers have been able to feed many more people per acre. Now, this has almost doubled with the introduction of precision agriculture in the 1990s. And we would need to double this again over the next 50 years to feed a population of about 10 billion. Now, this is possible as farmers can produce more food thanks to ongoing advances in genetically modified plants and the emerging high throughput technologies that are really accelerating the discovery of new microbial products. Next slide, please. The next agricultural revolution in the 21st century should include expanding the use of crop microbiomes to improve plant productivity. It is clear that crop yields must continue to increase to balance the increasing global demands for food and food security, which are continually threatened by climate change. Now, on a positive note, applying microbes to seed or fields can lead to interactions with plants that can increase yield and mitigate plant disease. Next slide, please. So a few definitions here. The microbiome is defined as a community of microorganisms in a particular environment. For example, the crop, plant, soil, or gut microbiome. The phytobiome is the community of plants, microbes, and animals, plus the geophysical environment. Now, under optimal growth conditions, where plant and soil microbiomes are interacting harmoniously with their environment, plants can be abundant producer, as you can see in the top soybean field picture. But the smallest change in biotic and abiotic conditions can tip the balance towards poor growth with extreme perturbations resulting in crop failure, as shown in the bottom picture. Can I have the next slide, please? Okay, the application of microbes to improve crop yields has a long history, all the way from growth-promoting rhizobia to fungi that provide protection from insects and drought. Other microbial products are useful to help crop plants take up nutrients or reduce disease. However, there are still many more areas to explore, including numerous untapped bacterial and fungi. And I want to add here also the microbial dark matter that's hidden to us. And there's also many challenges that we need to overcome. Among these is improving efficacy and to reduce year to yield and field to field variation, to scale up from small pilot studies, and to improve formulations to reduce costs to the farmer. Now, this can be achieved with a better understanding of the complex interactions of the phytobiome, along with current advances in technology. Next slide, please. As you all know, soil is a rich reservoir of microbes. A gram of soil is estimated to contain up to 10 billion bacterial cells and may hold as many as 10,000 bacterial species. Now, the number and diversity of bacteria, archaea, and fungi depend on the composition, acidity, moisture content, and other physical or chemical properties of the soil. Soil characteristics can also be altered by microbial activities. There's a cool video of bacteria moving along the fungal highway on the left. Next slide, please. Microbes and microbiomes can have both positive and negative effects on plants, depending on specific microbes present, as well as other factors, including environmental stressors. Some positive effects on plants include warding off pests, reducing the impact of pathogens, increasing a plant's ability to access nutrients, or surviving stressors, such as drought. On the downside, some microbes are pathogens, causing plant diseases with huge losses. Now, the challenge that we all face is to figure out how microbes respond to different stressors and how they interact with plants. And on that note, I'll pass it over to Megan Andrews, who will continue.
Great, thank you very much, Ignazio. So you may ask, why are we emphasizing the need for the microbiome-based solutions now of all times? And there's a number of factors that are involved that really makes now. I think I'm back. The right time to really focus on the microbiome as a way to develop solutions for agriculture. So one, as Ignazio has just gone through, we have a record of past success. There have been, over the past few decades, numerous microbiome-based, and we're talking about natural microbes, so organisms that are found in the soil that we can then use to add back to the soil and have a positive benefit for our crops. We also have, based on new technology and advances in science, we have a new understanding and appreciation for just how complex and dynamic the soil is. And when we look at both the microbiomes, whether it's crop microbiome or soil microbiome, and the phytobiome, so incorporating all of the geophysical attributes and the plant and the microbes, it is an incredibly complex dynamic system. Every organism is interacting with other organisms. And in order to truly use the microbiome to our benefit, we need to be able to understand how these dynamic interactions work. Now, what makes it possible to do this are advances in technology. So if you can imagine, if the soil is full of thousands or billions of different organisms, trying to understand how one particular microbe interacts with a plant, that's complicated enough. If you want to understand how a microbe interacts with fungi and the plant and the soil and the season and climate change, the amount of data becomes really quite staggering. So one of the ways to address this is to create interdisciplinary research fields where numerous different areas are working together to try and tackle this complexity with our new technology. So one quick example of this is NC State's Plant Science Initiative, where they are deliberately increasing research funding and infrastructure for projects that include plant improvement, data-driven ag bioscience, and resilient ag systems, and combining both academic and industry research. So this is just one example of what we see as the future of the microbiome in agriculture. Next slide, please. So as I said, we have new technologies now. So what do these new technologies allow us to do, and why is it important in terms of tackling the microbiome in agriculture today? Well, now we have the ability to not only look at who is in the soil, so looking at sequencing and analyzing the DNA, we also have the ability to tackle what they are doing in the soil and hopefully get to how they are doing it in the soil. So what are their metabolic products? What do they eat? What do they excrete? How do they interact with other microbes? How do they interact with the plants? If a drought happens or a flood happens, what is that sort of chain effect that goes all the way through the soil, through the microbial communities, and into the plant? That will ultimately affect productivity. So with all of these new technologies, we have the ability to look at soil in very small sample sizes, very large data sets, and really try to understand, and importantly, to predict how these interactions will happen. This allows us to then modify the dynamic processes and feedback loops that are controlling what's actually happening in a given field to our benefit. Ultimately, the goal is to increase plant health and crop yield through this management of the microbiome. Next slide, please. Now, similarly, advances in technology in the field are also allowing us to collect enormous amounts of data at a very detailed scale. So these include things like smart tractors, microsensors, drones and satellite imagery, and digital phenotyping. So one example here in this image is being able to take a drone up and take an image of a field, and you can look at this particular image and tell what parts of the field are healthier and what parts of the field are not, looking at the plants. Now, ultimately, what you want to do is be able to correlate that to some either nutrient deficit in the soil, a microbial deficit in the soil, etc. So this will allow you to then look at a field and say, this part of the field is doing well, this part of the field needs, we hope, a microbial application to bring its productivity up to an acceptable level. So the big challenge here, and what we see as really the next step in this particular field, is being able to correlate this complex microbiome data to the plant, soil, climate, weather, and land management data to be able to help improve agriculture through these microbiome-based solutions. So if you can imagine, each of these particular analytical or technology can create enormous amounts of data. But how do we use that data? That is really the next big challenge, and that is going to require interdisciplinary tackling from scientists, social scientists across the academic and industry spectrum. Next slide, please. One of the other things we really wanted to emphasize through this CAST publication was that it's not just supporting and investing in the research itself. It's beyond just this research. We want to create a foundation for success. We know that feeding the world and being able to increase our crop yields is a problem that we need to tackle over the next few decades. But what we can do now to make our chances of success even greater are creating a foundation for this interdisciplinary research. So this includes things like interdisciplinary training, developing collaborative communication strategies, and developing new research and applied partnerships. Next slide, please. So as some examples of this, we emphasize the need for interdisciplinary research. So this includes both funding and infrastructure that allows scientists, social scientists, and other relevant parties to collaborate and work together to solve these complex systems. So this can include people spanning analytical chemistry, bioinformatics, communication, economics is critical when you're talking about agriculture, environmental science, mathematics, and of course, microbiology, plant biology, soil science. So in order to have expertise in any one of these fields, that's great, you can solve that part of the problem. But in order to tackle the dynamic complexity of this system, you need to be able to both communicate with and collaborate with scientists and social scientists that span this level of expertise. Next slide. One of the other things we wanted to emphasize is that collaborative communication for successful commercialization of microbiome solutions is really critical. And what we mean by this is not just training scientists to be better communicators, although that's obviously part of it, but it's not just sort of top-down communication. What we're advocating here is collaborative communication to develop the best way to impart both the actual technical knowledge that we need to get across to growers, to stakeholders, to policymakers, but also to get feedback. What is it that they need? What is it that they want to know about these potential products? And really, how do we best communicate both the benefits to the grower, the benefits to the environment, and set realistic expectations for these microbial products? So all of this together requires scientists with the technical knowledge. It requires social scientists with knowledge of how to communicate across these different sectors. It requires policymakers and policy developers to be part of this conversation. So it really is a collaborative way to develop a strategy to better communicate microbiome-based solutions for agriculture to growers, to buyers, to consumers, to other scientists. So it's a wide range and maybe a new take on trying to get the information about these products out to a broader audience. Next slide, please. Now, along the lines of commercialization of microbial products, this is something that takes a long time. It is not something that happens overnight. There are multiple steps, including analysis in the lab, which can go relatively quickly, especially with some of the new technology that we have. But before it gets to the field, it also needs to pass greenhouse trials. It needs to scale up into field trials. And microbes are maybe not as predictable as we would like them to be sometimes. So something that can be very successful in the lab may or may not be successful in the greenhouse. Things that are successful in the greenhouse may or may not be successful in the field. But in order to bring it to a commercial product, it has to be successful in the field. So one of the things that we see as a very promising arena into bringing all of this technology together is finding a way to identify more cost-effective screening at earlier stages to better predict which successful microbes in the lab will translate to success in the field. That will help bring costs down. It will help reduce the time of discovery to market, and overall will make microbiome solutions a more viable and more successful solution to feeding the world. Next slide, please. As part of this, we also see partnerships between academic researchers and the ag biotech industry as really critical for taking laboratory success all the way to commercial success and growing success. Now, one of the tricky things here, it's not just about funding this type of research, although obviously that is important as well. It's also about decreasing the barriers and increasing the incentives for all parties involved. So this includes things like protecting intellectual property adequately, but also allowing academics to publish and to get adequate credit for their work in this arena in terms of getting tenure, in terms of getting their PhD, and other sort of milestones in the academic realm. So based on our experience at NC State, at least, the university technology transfer offices can really function as a critical bridge between industry and academics and help lower some of these barriers and help make very equitable incentives across the board to help encourage active and early collaboration between academic and industry scientists. Next slide, please. So we've covered a lot of bases today. Ignazio went through sort of the setup for why it's important that we increase yields using microbial inputs. There are a number of different factors that include very interdisciplinary research, all the way through these industry academic partnerships, increasing our communication, etc. So this slide really just sort of sums up how we see the future. what we need to do to be successful in using the microbiome to generate agricultural solutions. So on the outside, it's really the sort of human aspect, new graduate training models. So this gives graduate students a more interdisciplinary take, deep expertise in their field with the ability to communicate and collaborate with others. This will lead to increased interdisciplinary research, as long as it's supported with adequate funding. This will also help lead to more academic industry research collaborations. And of course, as part of all of this, we need better collaborative communication strategy development. Now, the next level in is looking more at the technological and research side. So if we can get the human component more interdisciplinary, more collaborative, working together to solve this problem, that will also lead to increases in new technologies, new advances in the area of phytobiome and microbiome research, and increases in big data, machine learning, other sort of technologies that allow us to both process and analyze these enormous data sets that are being generated. All of this together has the potential to lead to microbiome solutions for global crops, which is the goal. Next slide, please. So taken all together, the task force involved in developing this CAS publication came up with 6 recommendations that we see as setting the foundation now and investing where we need to invest to use the microbiome and have success in generating these solutions that we need to better our agriculture going forward. So these recommendations are to commit resources to understanding the dynamic complexity of the phytobiome through both research funding and infrastructure development. We suggest that funding the development and application of cutting-edge technologies, so things like machine learning, precision agriculture, development of microsensors, etc., any of those that are relevant to agriculture microbiome research should be funded. And specifically to take these areas that maybe machine learning has made progress in its own field, but we need a way to apply it to agriculture. And it's those sort of collaborations that we are really suggesting need to be developed. Recommendation 3 is to create initiatives to bring scientists, social scientists, and stakeholders together to develop strategies and best practices for effective science communication, public engagement, product regulation, and policy development for microbiome-based solutions. And that covers a lot, but we really want to emphasize that this needs to be a collaborative effort across the field. Recommendation 4 is to increase the funding and incentives for interdisciplinary research, education, and training in the agricultural and microbiome fields. To encourage new models for training graduate students to increase their cross-disciplinary literacy, their ability to work as part of a team, and their communication skills, especially outside their very specific expertise. And last but not least, we recommend that everyone should encourage crop microbe-focused industry academic partnerships and provide programs and funds to help bridge the gap between promising laboratory results and successful field trials. So hopefully the sum of these recommendations has made it very clear that the microbiome field has a lot of potential in terms of developing solutions for the agricultural field, but it is also attainable. But one of the important things is that we need to start investing in this now. Investing not only in terms of funding, infrastructure, programmatic shifts, and appropriate incentives and barrier reduction across the field. So with that, I think I'm going to hand it back to Kent, and thank you very much for all of your attention.
Thank you, Ignazio and Megan, for that excellent overview of the highlights and key information of the paper. We are receiving several questions that we will begin to address with our panel of subject matter experts in just a couple of minutes. As we prepare for those questions, let me share some information with you about some of our upcoming publication releases. Next slide, please. The next paper we're releasing is commentary, the importance of communicating empirically based science for society. We're released on September 21st. We will be followed by a webinar on September 22nd. And the task force chair, Stuart Smith, University of Saskatchewan, will share the highlights. In mid-October, watch for the issue paper, Food Biofortification, Reaping the Benefits of Science to Overcome Hidden Hunger. with the chair, Dr. Howard Boyce, with CGIAR. And then in November, we'll be releasing another issue paper, Ground and Aerial Robots for Production Agriculture, Opportunities and Challenges, with the chair, Dr. Santos Pila, University of Nebraska-Lincoln. So please stay tuned and watch for those upcoming releases and webinars. Next slide, please. Once again, please let me welcome back our presenters and also introduce our panelists. Joining us for the Q&A session today, we have Dr. Ignazio Carbone, Dr. Megan Andrews. We're also being joined by Dr. Elizabeth Stulberg, with the Science Policy Manager with the Alliance for Crop Soils and Environmental Science Societies, Dr. Gregory Maloney, Senior Scientist with Novozymes BioAg, and Dr. Carolyn Young, Professor with the Novo Research Institute. Thank you all for being here with us today. They're all members of the task force that helped contribute to this. Now I'll turn the program back to Megan to kind of lead our Q&A session for today. Megan?
Great, thanks, Kent. So we actually have quite a few questions. This is great to see how involved the audience is. I'm going to go through and we'll see how many we can get to. So first question, I think this is going to be for Carolyn. Question is, from the top of your presentation, per capita per cropland acres doesn't necessarily mean the absolute land or the absolute amount of ag land is decreasing. Based on what we see happening in places like Brazil, so cutting rainforest, etc., it could be that absolute cropland is actually increasing. We're just seeing rising population driving the need to use those acres more productively. Carolyn, can you comment on this?
Yeah, so I would say we are seeing a rising population driving the need to use acres more productively. We're seeing city sprawls. I'm based in Oklahoma, just north, not far from the Dallas area, and we've got some statistics around that area where that community is sprawling out and it's taking over some of the good cropland and grazing lands and forcing people to go out into less productive areas. So people have to be more productive with a less productive area. So they are some of the challenges that we need to overcome.
Great, thanks, Carolyn. Next question is talking about looking at the notion of changing the microbiome in a field. How long lasting is this effect? And is a new application of a particular microbe needed every year, or would a large dose permanently, or at least for several years, change the phytobiome in that area? So Greg, this sounds like one for you.
Sure, thanks, Megan. And it's a great question, and it's one that we think about a lot when we're developing products in the microbial ag industry. But one thing we do see typically is that microbes that you apply as a product to a field do not stick around for more than that season. So yes, you typically need to apply year after year. And the main driver for that is that usually the microbes you are applying are not one of the native dominant microbes in that field. They could be native to that field, but you're most likely putting in a different strain of that species. And it's just usually not going to kind of take hold because it's going to be competing with everything else there. The other thing is that usually when you're putting a microbe in a field, a microbial product, that product is one that's been chosen to grow well or adapt or colonize in the presence of a plant, whether it's the root or the foliage or some part of the plant. And once you harvest that plant and take it away, that's when it really starts to decline and the native microbiome comes in and sort of takes over again. With that said, there is some efforts in the field. There's actually a lot of efforts in the field to identify microbes that maybe they don't stick around forever, but once you put them in the field, they can change the microbiome, especially the microbiome colonizing the rhizosphere. So we can sort of use microbial products as prebiotics in a way to alter the rhizosphere microbiome to be more beneficial to the plant.
Great, thanks, Greg.
You're welcome.
All right, so the next question, this is going to be for Elizabeth. Do these products fall under the umbrella of GMOs/ag biotech? This is a great question. The concern is, if you speak from a global standpoint, many nations do not allow commercialization of genetically modified products. From an economic perspective in the US, the costs of approval and regulation can be quite a bit higher. Or does this fall outside the realm of genetically modified ag biotech subject to the biosafety regulations? And then sort of linked to that is, how can scientists work to harmonize the regulation of plant biologics internationally?
Yeah, this is actually a really great question. Thanks so much. Right now, I think the things that we're really talking about in this paper do not fall under the umbrella of GMOs. We're talking about taking microbes that already exist in nature and not necessarily changing their genetics around, but putting them potentially in new combinations, or even combinations that they already exist in one place and bringing them to another place. So GMO regulations wouldn't necessarily apply. I mean, it might eventually, but they don't, strictly speaking, what we're talking about right now, they don't necessarily apply. But there are regulations that would apply. For example, if you're talking about a microbe or a combination of microbes that acts as a pesticide, EPA might get involved because you're using it as a pesticide. And then a biostimulant is another example. If you're using one organism or a collection of organisms to change the growth of a plant in a specific way, again, you might encounter regulations that yes, could take costs of approval to get through, or to demonstrate that it really is safe, and that could raise the barriers for being able to use this technology if there's a lot of testing that needs to be done. The second part of the question, or I guess the second question, has to do with harmonizing regulations across, or I guess internationally. And yeah, I don't think I have a very good answer to this second question. How you harmonize regulations internationally is something USDA's Foreign Ag Service and the State Department have like whole teams of experts and ambassadors to try to achieve. And it's definitely something that they're working on. And with this new realm of possibilities, I hope very much that we achieve some kind of harmonization. That being said, I doubt very much that this, famous last words, I doubt very much that this will be a trade issue in the same way that GMO crops might be a trade issue. If you're using microbes as a treatment, or for example, in the soil, and you grow crops, it definitely won't fundamentally alter the genetics of those crops. So I doubt very much that we would see some sort of a trade issue that you would need harmonization for in that respect. So I hope that answers those questions.
Great, thanks, Elizabeth. That's definitely much better than I could have done. It's a very complicated situation, for sure. But that is one thing that I think Elizabeth really touched on, is that what we're focusing on in terms of the microbiome is not genetically modified. It's just using the naturally occurring microbes in new ways or new combinations. So next question, Ignazio, this one's for you. How much do you think priority effects from the natural microbial community impact the variability between trials? And specifically, do you think you can control for within community interactions? So I know that's a complicated question, but just very briefly.
Yeah, that's a great question. It's complicated, and I think Greg touched upon it, the native versus introduced biologics and how they perform. It is possible that a single organism can have a transformational effect on the microbiome, and we've seen this with a few organisms. It's just difficult to say what other interactions, because we often study one organism at a time. Studying multiple organisms, particularly across different scales, by that I mean individuals, populations, and then closely related species makes things even more complicated. But yes, the indigenous environment does play a huge role in how equilibrium reestablishes after a strain is introduced. But we know very little about the community interaction so far. We need more long-term studies and trials over multiple growing seasons to really get a handle on this.
Great, thanks. All right, so I think next question is going to be for Greg. When and how to involve industry in the research timeline? What would you suggest for scientists just starting a microbiome research project? This is a really great question.
Yeah, it's a great question. Hopefully I understand it correctly. Is this somebody coming from an academic field wanting to partner in industry? Is that
I think so.
Is that what you're getting, Megan? Okay, yeah. There's a few different stages at which you can, at which it's a good time to go to industry and pitch an idea and try to partner. I think the best time to do it is when you have significant data to back up your product lead or your idea or your technology. It really helps to have field data to really drive home the point that you have shown a realistic beneficial effect of the microbe or active that you want to partner on. On the other hand, if you can be in the stage of just sort of having an idea for how to screen for a new microbial product, if you have a novel screening capability, that would be something of great interest to industry. So it sort of depends. You can come in really early in the product pipeline where you don't even have a microbe selected or a group of microbes, but you just have a technology for sourcing them, or you can come where much later in the pipeline where you have maybe just a handful or one active or strain that you think and have data to back up is worth commercializing.
Great, thanks, Greg. And that's definitely been our experience at NC State through the Center for Integrated Fungal Research. There's been a mix of very early interest with technology or developing methods, or later interest where you actually have something that you think has potential to be a product. But it's definitely beneficial to reach out if you're interested in forming those partnerships. You can always have early discussions under non-disclosure agreements, so there's no real risk, and it's worthwhile for sure.
Yeah, it can't hurt to reach out, never.
All right, so this next one will be for Carolyn. So we have a sort of question/comment. That they heard a strong bias towards microbial products as the targeted deliverables of this research. As part of the comment, they are sure they have a role to play, but are concerned that microbial, quote, magic bullets have limitations just like chemicals and other land management tactics that we have now. They're more intrigued to find out how cultural practices may shift natural microbial communities towards the beneficial versus just adding a single strain or a few strains to hopefully have an impact. So Carolyn, can you sort of tackle that?
Yeah, I can. So I think this is a great comment, and there are a couple of other questions that have been posted that have also sort of aligned with this approach about how are we trying to be very microbial product driven, where we're adding something to the soil rather than trying to manage what's already in there. There's a lot of research and focus that's coming on soil health now and regenerative agricultural practices, and using all of the same methodology that we would use to understand what's in the microbiome and identifying key players in the microbiome may help us actually understanding how these regenerative ag practices and these soil health practices are helping to change the microbiome communities as well. And I think this moving on will help us see that, yes, maybe we can help regenerate the soil and have better cultural practices, moving these microbial communities towards more beneficial microorganisms. So there's probably a place for both. There's definitely some transitions. There are definitely some examples where the single microbe, such as a rhizobia, will help with the nodulation and nitrogen fixation with legumes, for example. So that would be something that would be a practice that could continue. But taking on board these new cultural practices and thinking about ways with soil health practices, keeping the soil covered, trying to keep the plant diversity up there, trying to minimize the disturbances of the soil, and that includes reducing some of the inputs such as the fungicides and things like that, may actually help us to manage some of these systems a little bit better. But we need the technology to make sure we understand that that is what's happening.
Great, thank you so much, Carolyn. So sort of, I guess it's almost a corollary to that in terms of trying to understand and generate the data that we need. Here's a question for Elizabeth. Would it be wise to establish some sampling, analytical variability, and statistical assessment of the data gathering effort? If we could establish confidence levels for each input, we may be better able to focus resources where high variability limits our ability to fully utilize data.
Yes, that was an easy one. Standardized sampling would be terrific. I would love to see a unified, nationwide or maybe even international data repository. Right now, for example, NIH hosts a data repository for genetic information, right? NCBI hosts a great database that people can, you put in your genetic sequencing information, you can search against everything that's been found. I wish USDA would do this. I have written loads of comments and suggestions to USDA. Please do this. The private sector hasn't been able to pull this together. I think we would all have loved to see, some great competition, the private sector trying to make, databases that that their customers could use, that scientists could use. The problem is that most private sector companies that would benefit from having this database also have their own proprietary data that they don't necessarily want to share. So it leaves researchers, it leaves farmers in kind of a bind with these non-interoperable databases with data that they don't necessarily know how to really use to their benefit. So I'm really excited about the idea of creating what's called a FAIR database. FAIR stands for findable, accessible, interoperable, and reusable. And it to me, it's something for the government, for USDA to put together standards. So, a UAV flying over collecting data would just automatically put it in that format. A farmer, a researcher could choose whether to upload it into this national database, keeping in mind that researchers already often have to, if they get federal funds, they have to upload their data somewhere. But where they upload it is not necessarily any place useful. The data can't be reused. It's not in the right format. But just imagine a database where, if a researcher wanted to know whether there's like a connection between bacteria or microbes in the soil microbiome in like a given geographical area and the risk of a crop disease, you could just look. Maybe there's like a map. Or what about how the microbiome changes after a natural disaster, like a wildfire? Or you could layer on epidemiological data. What's a person's risk of getting a stroke or cancer based on the soil microbiome that they, where they grew up? You could just look into this database and it would already include all of these things. I think the sheer cost of doing this research would go way down. So that's one of the things about microbiome research, just the sheer amount of data and the overwhelming need to have that kind of a standardized database oh, it has to happen. It's going to happen. I'm so excited about it happening. I really can't wait for it to materialize.
Thank you so much. That's a great answer and a fantastic question. So I'm going to tackle a couple really quick because I think they're super critical and they're sort of related. So question number one is, how do we ensure the public is kept up to speed to avoid the current EU precautionary principle rejection of extremely important science accomplishments? And sort of going along with that, there was a question about the extension network at NC State is really large and connected to growers. Are we using them as a resource to connect to local farmers? So the easy answer to that is yes, absolutely. That is a goal. The extension network at NC State is very large. It's very connected, but that really only applies to North Carolina. This is something that we need to look at in a broader sense in terms of the communication. One quick example at NC State is they have recently hired a professor who is specifically in a microbiome extension position. So NC State really is sort of, along with the Plant Science Initiative, they're trying to sort of take the lead and really emphasize the importance of the microbiome in agriculture and how to take what we're learning at the university level, and getting it out to growers, et cetera. In terms of the general communication, I think this is one of the reasons why we're really trying to emphasize in this paper the need for collaborative communication strategies. So we want to avoid the situation, where the EU is just rejecting, I believe, a lot of the CRISPR stuff just because they don't, out of an abundance of caution, they're being very conservative. We also want to avoid sort of the backlash that some of the GMO has had. So the only way to do that is to be proactive and to be collaborative. This can't be something where scientists just say, this is new technology, this is great, it's going to solve all of our problems, please use it. Or we just assume we will use it. This needs to be a bigger conversation so that we can get that information broadly disseminated in a way that effectively gets the information across and allows a thorough discussion to happen at every level, from academic science, industry science, growers, people buying their groceries in the grocery store. Like, all of those levels need to be brought together to effectively communicate this. So I think we have time for just a couple more questions. There is a question about, are we also considering the possibility where a beneficial microbe is transformed to a plant pathogen? So this is part of the reason why we need to have the ability to understand all of the complexity, because I'm sure there are many microbes that under certain situations can have very great benefits and under other situations can act as a pathogen. But I think, Ignazio, maybe you could address this a little bit more.
I don't have any examples of that. I mean, no, I don't have any examples where that can occur. It's possible, but I'm not aware of any.
Okay. And Elizabeth, I think you wanted to mention along those lines, sort of clarify that maybe the beneficial microbe is not genetically changed into something that's a plant pathogen.
I think I have to agree with Ignacio. I know of no examples where you could take a beneficial and put it someplace new and it becomes a pathogen. That happens in human medicine, where you have a perfectly harmless E. coli, but put it in the wrong place and it can make you sick. I can't think of any off the top of my head examples of that with plants. And the question had to do with like transformed into a plant pathogen. That word transform usually means genetically modify. So if we're talking about, genetically modifying these microbes, no one's really doing that yet. So I'm not sure why you would want to transform it into a plant pathogen. I think USDA would have something to say about that. In fact, USDA APHIS would regulate it under its Plant Pest Authority. It would be highly regulated. You need a permit to use it. That being said, if you're taking a beneficial microbe and you discover that it does turn into a plant pest when you move it into a new place or have it in a new situation, even without being genetically modified, USDA would regulate it, and you would need a permit to use it, just like a chemical.
Great, thank you. So it sounds like that's at least less of a possibility than maybe in human medicine. So that's good news. So we have a ton of really good questions. I think we're gonna go one last question for Greg. How much potential do microbes or microbial consortia have to increase crop yields compared to yield increases from crop breeding?
Yeah, I think this is another great question, and it kind of gets into what microbial products can actually achieve. I know somebody mentioned the word like magic bullet or silver bullet, and it's really not what we're typically aiming for with a microbial product. What we usually aim for is something that can be complementary to plant genetics, can be complementary in a way that if a plant which is bred to achieve its highest yield potential encounters some stress, whether it's nutrient stress or water stress, pathogen, anything like that, the microbe is more of a sort of an insurance policy. It's actually, it's pretty common in a field. You don't have crops which reach their full genetic potential. And if you could get there without a lot of expensive or inputs or synthetic chemical inputs, or if you can even just reduce the amount of fertilizer or pesticides you're applying on the field by applying microbes, we consider that a win. So I guess in short, they're kind of separate. We don't expect that microbials will go above and beyond plant genetics. They're sort of there as an insurance policy for when the plant can't reach its full genetic potential. That's the way I look at it, and we're not looking for a silver bullet. We're really just putting microbes there to alleviate different stresses in a lot of cases.
Great, thank you so much. I just want to take a moment to thank our panelists. You guys have been really great in answering questions. And I also want to thank the audience for all of the really great questions. I know we didn't get to all of them, but thank you so much. And I'm going to hand it back to Kent.
Thank you, Megan and Ignazio and all of the panelists. Thank you very much for your work on this paper, as well as your fantastic job of sharing the highlights and the question and answer. As Megan noted, we didn't get to all the questions. We will summarize those questions. We'll share them with our panelists and look for the complete set of answers to be posted on our website or with the paper in the next week or so. So we'll keep you updated about that. If you've not accessed and seen the paper yet, you can get that on our CAST website. Here is the address to find that. Again, our papers are free to download. We would encourage you to do that, as well as look at the executive summary, the quick cast, the study guide, the other resources that are as a part of that. And also, we've included here the email addresses of our panelists today if there's any particular things that you would like to follow up with them specifically about. And again, I'd like to echo what was said earlier. Thank you for being a part of our webinar today. Thank you for your support of CAST and our efforts to communicate unbiased, credible, science-based information. We hope you enjoyed it and found it very informative. Please be watching for upcoming information about our future webinars that we talked about earlier today, and hope you'll be able to join us. I'd also like to acknowledge and thank our friends at Dairy Management Inc. for their assistance in helping us provide this webinar. Thank you and have a great day.